Hello friends, this video on molecular basis of inheritance part 9 is brought to you by examfear.com. No more fear from exam. So now the focus was on finding out the biochemical nature of the transforming principle. So now regarding the biochemical nature of the transforming principle. So transforming principle was the name given to the substance which is getting passed on from one bacteria to the other bacteria. So that substance was called transforming principle by Griffith. Now in order to know the biochemical nature of the transforming principle, somebody thought that it could be protein. Some of them thought it could also be fats or carbohydrate, or it could be nucleic acid. So there were different opinions that what it could be. So bacteriologists performed a series of experiments to identify the transforming principle. So some of the experiments which they performed were, they precipitated the transforming principle with alcohol. And it was found that the transforming principle was precipitated when alcohol was added to it. So what did this prove? This proved that it was not a carbohydrate, because had it been a carbohydrate, it would have not precipitated with alcohol. So these are some of the properties of various different biomolecules, like the properties of carbohydrates, proteins, and I hope you are aware of them. Again, they also used the protein digesting enzymes called proteases. So they found that the transforming principle could not be destroyed with proteases. When I say destroyed, it means that, so for example, proteases are the enzymes which digest the proteins. So if you mix protease with proteins, what will happen? The proteins will get destroyed. Destroyed in the sense the proteins will get digested. But it was found that the transforming principle could not be destroyed with the proteases. What does this prove? This proves that the transforming principle was not even proteins. Because had it been proteins, then the protease would have digested the transforming principle. It was also found that the transforming principle could not be digested or could not be destroyed with lipases. So that means they were neither lipids. So they were not carbohydrates, they were not proteins, they were not lipids or fats. So what were they? Now these transforming principle then looked like little similar to nucleic acids. That is, they might be rich in nucleic acids. But again, in nucleic acids, there were two options. One is RNA, the other one is DNA. So what could be that? So then it was tried with the enzyme that digests the ribonucleic acid, that is RNA, and that was ribonuclease, and it was observed that the transforming principle was not inactivated with ribonuclease. So what did this prove? This proved that it was not RNA. So now the only option that was left over was the DNA. So they found that when they used deoxyribonuclease, it could be inactivated, proving that it was DNA. Because when the DNA digesting enzymes were added to it, the transforming principle was inactivated. So that means it was DNA. So this is how it was observed that the genetic material, or the material that passes on from one organism to another organism, that is nothing but DNA. So DNA is the genetic material. So this was finally proved after Griffith's experiment, when Griffith defined transforming principle, and finally the biochemical nature of transforming principle was known to be DNA. Thank you. Please visit www.examfear.com to watch more videos, attempt free online test, get free study material, find tutors and mentors. Thank you once again.